Hello, my name is Norfitria Benti Ahmad Munir and my matrix number is 23DBT20F1036. Today I want to show you how I perform the four lab simulation, which are gram staining, catalase, coagulase, and lastly oxidase. First, I want to show you the lab simulation of gram staining. So we can start. First, hit the loop and take the bacteria from a liquid culture. Then put in a drop of water on slide. Next, heat the loop to sterilize it. So wait for 5 minutes to let water dry on slide. After that, use the Bunsen burner and pass the slide gently 2 or 3 times through the flame, and do not overheat case this will distortion of the cell. Okay, done. Okay, so next, flood the slide with crystal violet for 1 minute. Then rinse with water. Next, flood the slide with iodine for 1 minute. Then rinse again with water. Next, decolorize with alcohol for 5 to 10 seconds. And don't forget to rinse with water again. Next, flood the slide with safranin for 1 minute. Then rinse again with water. Lastly, view the slides under the microscope. So this is the result in the microscope. So the color purple is the gram positive. This one is gram positive, and the pink color is gram negative. So for the second lab simulation is catalase task. First, drop the hydrogen peroxide on slide. And pick the isolated colony with toothpick. First, we use the Streptococcus pyogenes. Okay, then mix the bacteria into hydrogen peroxide. Place the tip of the stick over drop, then click and drag to stir. So there's no bubbling, and the result is the catalase negative. Don't forget to dispose of the toothpick and dispose of the slide into the biohazardous waste container. So next, repeat the step with the second isolated colony, which is Staphylococcus aureus. So drop the hydrogen peroxide on slide and pick the isolated colony, which is Staphylococcus aureus. Then mix the bacteria into hydrogen peroxide. Place the tip of stick over drop, then click and drag to stir. So we can see that Immediate strong bubbling was produced. So the result is the catalase positive. Dispose the toothpick and dispose the slide into the biohazardous waste container. So for the third lab simulation is coagulase. So start. First drop water on slide. Then pick the isolated colony which is tough areas with toothpick. Then mix the bacteria into water. Don't forget to dispose the toothpick to prevent the contamination. Next, add 1 to 2 drops of rabbit plasma and rock slide and watch for clumping. So the result have clumping and this is the coagulase positive. So dispose of the slide into the biohazardous waste container. Next, we repeat the steps with the other of isolated colony, which is tough epidermis. First, drop water on slide. Next, pick isolated colony, which is tough epidermis with toothpick. Then mix the bacteria into water. As usual, dispose of toothpick. Next, add 1 to 2 drops of rabbit plasm. Then rock the slide to watch for clumping. So for this is does not have the clumping and the result is the coagulase negative. Don't forget to dispose of slide into the biohazardous waste container. So for the last lab simulation is oxidase test. So start the lab simulation. First, pick an isolated colony with a sterile toothpick. For this lab simulation, have 3 isolated colony, which is Salmonella enteritidis, Pseudomonas aeruginosa, and lastly Citrobacter frendi. So first pick on isolated colony Salmonella with a sterile toothpick. Then rub bacteria onto slide and watch for color change. Okay. So the result is oxidase negative. because there's are no color change. So don't forget to dispose of toothpick. So next repeat the steps. Pick on isolated colony Pseudomonas aeruginosa and rub bacteria onto slide and watch for color change. So for this is the oxidase positive because the color is change. So dispose of toothpick, then repeat again the steps. Pick on isolated colony Citrobacter frindi, then rub bacteria onto slide and watch for color change. Okay, so this is the oxidase negative because there's no color change. So don't forget to dispose of toothpick and dispose of dry slide. So done.